fingerprinting. DNA fingerprinting, I told, that is the Alex Jeffrey, and he is considered as a father of DNA fingerprinting. So this technology was first evolved by Alex Jeffrey in 1985, developed or evolved by, so same things. Okay, so the technology was first developed by the Alex Jeffrey in 1985. It is technology used to the identification of the individual at genetic level. Okay, if suppose that two person is the same in figure, same is the structure, everything will be the same, but you can't identify whether both are the same or not. So then for this you can perform the DNA fingerprinting. With the help of DNA fingerprinting, you can find they have the difference between genetic label, the individuality at the genetic level. So this is generally performed by the DNA fingerprint. Now, okay, DNA fingerprinting involves identifying difference in the specific regions, that is called the sequence, and this is called the repetitive DNA. This is called the repetitive DNA. What is means of repetitive? Means suppose that A. If in the DNA molecules they have like this sequence, so this is called the repetitive DNA. A repetitive DNA means the base pair is the same. So they have the DNA fingerprinting involves identifying the difference in a specific region. Okay, specific region, and this region have the repetitive DNA. Suppose that DNA length, 2.2 meter is the length of DNA molecule, 2.2 meter. And in this DNA molecules, what you have observed, some places they have the base pair, and this base pair is the repetitive type, like A, means this is the repetitive. So this can be used for the DNA fingerprinting because they have the unique features. Repetitive means unique. Okay. So these repetitive DNA are separated from the bulk genomic DNA at different peaks during the density gradient centrifugation. Okay. If you'll observe the density gradient centrifugation, this is the test tube. In the test tube, what you will observe, so they have the different sequences of the DNA molecules like that. Density gradient, I told already, cesium chloride density gradient centrifugation. Can you tell where I told this? Cesium chloride density gradient centrifugation. Which experiment? Very good, Gauri. Meselson and Stahl experiment, where he used the cesium chloride density gradient centrifugation. Okay. Now, so these repetitive DNA are separated from the bulk genomic DNA at different peaks during density gradient centrifugation. The bulk DNA forms a major peak and the other small peaks are referred as a satellite DNA. Okay. They have the two. One is the major, and bulk DNA forms the major peak and small peaks. Small peaks and a major peak. Okay. Suppose that this is the centrifugation. One is the small peak and one is the large peak. So small peak will be for the satellite DNA. So this you can say this is the satellite DNA. This is the satellite DNA. It is the satellite DNA. And large peak, that is the bulk DNA. It is, you can say, it is known as the bulk DNA. Bulk DNA. Okay. So they have the small, means whatever you have to learn, that is the satellite DNA, they have the small peak. Okay. Satellite DNA, it is the small peak. And we have to see already in chromosome. If you see the structure of chromosome, so they have the smallest structure and this is the largest structure. So this is structure also called the satellite. Sometimes you have seen a structure of the chromosome. Okay. So now these sequences show high degree of polymorphism and form the basis of DNA fingerprint. Who shows the high degree of polymorphism? Satellite DNA. Satellite DNA. Satellite DNA shows high degree of polymorphism. High degree of polymorphism means we can use for the DNA fingerprint. High degree of polymorphism means we can use for the DNA fingerprint. So this again, this sequence show high degree of polymorphism and forms basis of DNA fingerprint. The inheritable mutation is observed in a population at high frequency and it's referred as a DNA polymorphism. What is the DNA polymorphism? Means inheritable mutation observed in a population with the high frequency. What means of this? Suppose that, look at here, this is the DNA molecules. Okay, is the in this DNA molecules A, T, G, C, T, A. This is the normal DNA. Next time mutation takes place. A, T, Z, C, T, A. DNA mutation takes place, and this mutation is inherit. Okay, this mutation is inherit because I told mutation is the responsible for evolution. Raw material for evolution is the mutation. Clear? You already studied the raw material for evolution is the mutation. So here they have the inheritable mutation is observed in a population at a high frequency. Okay, means that there is the high frequency means too much mutation is there. So high frequency of the mutation is there, and this high frequency mutation is known as the DNA polymorphism. Okay, and DNA polymorphism, I told it is act as a marker. It is the basic marker for the DNA fingerprinting. Okay, or you can say it is the basic tool for DNA fingerprinting. Okay, DNA polymorphism. This phenomenon is play important role to DNA fingerprinting. Okay. The principle of DNA fingerprinting is based on the matching of VNTRs. What is the VNTRs? This is also very important. VNTRs, variable number of tandem. Variable number of tandem repeats. Okay, so VNTRs, this is called the variable number of tandem repeats. Okay, the principle of DNA fingerprinting is based on the VNTRs. Okay, means in the DNA polymorphism they have the particular sequence, and this sequence is known as the VNTRs, variable number of tandem repeats. Variable number of tandem repeats. So it is called the means it is the basic, it is the principle of DNA fingerprinting. Okay, and VNTR of DNA collected at the crime spot where the crime have happened. They will collect from the VNTR. Means they will isolate firstly DNA, then they have to observe the VNTR. Means directly they will not gain the VNTR. So DNA collected crime spot with the suspected person DNA. Okay, they have also the suspected person DNA. So VNTR is the principle of the DNA fingerprint. Now, VNTR, what is this? This is the very important variable number of tandem repeats. It is also called the Minisatellite. It is known the Minisatellite, microsatellite, Minisatellite. So microsatellite they have not given, so I'm not going to talk. But Minisatellite that shows a very high degree of polymorphism. Okay, in compared to microsatellite polymorphism. And what is the polymorphism means? The mutation which is inheritable into the individual. The mutation frequently mutation which is inheritable into the individual. So that is called the polymorphism. Okay. Now, so Minisatellite, they have the high degree of polymorphism. So that's why we are using the Minisatellites for the DNA fingerprinting. Okay, just wait. Now, VNTRs are very specific to individuals and differs from person to person. It shows some similarities between family members. Okay, it shows a little bit similarity, but most of them VNTRs are very specific to individuals and differ from person to person because variable number of the tandem repeats it have. It shows the polymorphism. It shows some similarities. It also shows the some similarities between the family members. Okay, they have the means. Most of the time they have the similarity, but little bit differences is there. And which is the basic or which is the principle of DNA fingerprinting. Now, VNTRs are identical twins or the same. If the identical twins you have obtained the VNTR, this will be the same. There is no variation because genetic content is the same. Identical twins, they have arrived from, they have born from same mother. So means their genetic constituents will not change. So VNTRs of identical twins are the same. Hence, it is not possible to identify individuality in the identical twins by the DNA fingerprinting technology. Very, very important and drawback. So very big drawback of the DNA fingerprinting technology. If the twins is the identical twins. Are you getting it? Twins, because different types, zygotic twins, fraternal twins, and identical. So in case of the identical twins, so VNTRs cannot differentiate. Okay, so variable number of tandem repeats it can't use for the identical twins. If fraternal twins we can use, zygotic twins we can use, but identical twins we can't. Okay, so this is the big drawback of the VNTR. So VNTR are very identical twins are the same, hence it is not possible to identify the individuality in identical twins by the DNA fingerprinting technology. Now, southern blotting, it is the principle. There is the different types of the blotting. One is the southern blotting, and another is the northern of the transferring DNA from agar gel to nylon sheath. This is the gel. In this agar gel, they have the DNA molecules, means these DNA molecules, they will present like that. These DNA molecules present like that. And we have to one important substance, which is known as the nylon sheath. When this RNA, sorry, when this DNA molecules transferring on the nylon sheath from the agar gel. Okay, the transferring of DNA molecules from agar gel onto the nylon sheath. Okay, so this phenomenon is known as the southern blotting. Southern blotting is clear. Southern blotting means you can say the transfer of DNA from agar gel. The transfer of DNA, transfer of the DNA from agar gel to the nylon sheath. That is called the southern blotting. If the northern blotting means the transferring of RNA from agar gel to the nylon sheath. And if the eastern blotting means the transferring of protein molecules from agar gel into the nylon sheath. Okay, so this have given about the southern blotting. Another is the probe. What is the probe? Just listen. Probe means you have to see if you are going to prepare the curd at your home. Most of the person they belonging from the rural areas. Okay, so at your home, mother, if she is going to prepare the curd, she is going to prepare the curd. Okay, if she is going to prepare the curd during if they have she is going to prepare the curd, what she will do, they have the inoculum. She has the inoculum. What is the inoculum? Inoculum is nothing but it is the only Culture of bacteria, lactobacillus, lactobacillus bacteria. Inoculum means if you firstly our mother, our house mother, she will prepare the curd. How? Firstly boil the milk, and when she boiled, after that they introduce the inoculum. She will introduce the inoculum. Clear? So similarly, inoculum probe is there. Inoculum is short quantity of the bacteria, little quantity of the bacteria. So this is called the inoculum. Similarly, inoculum probe is there. Probe, it is the also short stretch of the DNA molecule. It is not long total DNA length. It is the short stretch of the DNA molecule. That is called the probe, means initiator. Now here you can see probe, single stranded polynucleotide fragment complementary to a specific sequence of the nucleotide DNA. It is called the probe. Single stranded polynucleotide of the frag. Suppose that this is the DNA molecule, okay, and this DNA is the this is the 5' end, and here this is the 3' end. What happened? You have to probe. Probe is the short stretch of DNA molecule. So they have the small short stretch of the DNA molecule. This will be the 3' to 5'. So it will be the overlap, because probe, it is the complementary. It is the complementary of DNA molecules. Okay. A single stranded polynucleotide fragment. It is saying single stranded polynucleotide fragment complementary to specific nucleotide. Complementary to specific nucleotide, and this is called the DNA probe. It is majorly, or it is basically used into the identify VNTRs and desired gene. Okay, this probe generally used for which purposes? To identify the VNTR, where it have the variable number of tandem repeats, or desired gene where it is present. So with the help of probe, we can identify the location of VNTR and desired gene. Clear? Is it fine?
Yes, sir.
Okay, very good. So the basic function of the probe, it is the complementary of the polynucleotide strand, and it identifies the location of VNTRs and desired gene. Because whatever you are going to find, you have to already sequenced with the probe. Technology. How many steps is there? Firstly, you will take the leaf. This leaf cell sample, leaf crust, and with the help of DNA isolation techniques. Can you tell in DNA? I think sir already taught to you DNA isolation techniques. So DNA can be stained by which substance? DNA can be stained by which substance? Yes.
Yes, sir.
Can you say again? Now she is not saying. That is ETBR. This is called the ETBR, ethidium bromide, ETBR. Okay, so DNA stain. So cell sample extracted DNA. During the extraction, we generally use the chilled ethanol, DNA precipitate, and ultimately settle down into the base of the tube. Okay, DNA extracted with the help of enzyme, cleavage of the DNA by restriction enzyme. So restriction enzyme, okay, restriction enzyme, it is used to cleave the DNA molecules into small fragments. Okay, ultimately separation of DNA by the fragment electrophorese. Okay, through the electrophorese, different DNA molecule segment present. Okay, because DNA molecules, they are present the overall in the form of scattered, but when you have performed the electrophoresis, The DNA molecules can be separated on the behalf of their size, on the behalf of their density. Okay, the DNA molecules, which is the large, they moves very fast and it will go the down. And DNA molecules, which is the small, and they have the short stretch of DNA molecules, it will be the at the top. That is called the well. Here near this well, we generally use the DNA molecules. We load the DNA molecules. Now, southern blot means from agar gel, we are going to transfer onto the nylon sheath. We are going to transfer onto the nylon sheath. So this is called the southern blotting. Ultimately, binding of radioactive DNA probes to a specific DNA fragment. They will bind with this. And membrane washed free excess probe. So those probes which is excess, they are completely washed. X-ray film used to detect the radioactive pattern. So I will means we will use the X-ray, okay, and this X-ray we generally use for the detection of the DNA molecules. Okay, so plant A sample, plant B. Okay, this is the sample molecules means probe. This is the difference between sample A and sample B. Okay, so plant A and plant B. So this is the steps. So next is the DNA fingerprinting techniques involved. I told steps you have to learn it. So DNA fingerprinting techniques involved southern blot and hybridization using radioactive radio-labeled VNTR probes it included. So how many things needed? First one is the southern blot, second is the hybridization, and third is the radio-labeled VNTR. So three components are important for the DNA fingerprinting. Okay. So they have the 5 different steps. Just look at 5 to 6. Isolation of DNA. I told firstly extract the crush the cells, destroy the cells, and extract the DNA molecules. This DNA is the very big. So what you will do, you will digest the DNA molecules. Okay. So digestion of DNA by the restriction endonuclease. Restriction endonuclease enzyme, with the help of this you can digest the DNA molecules. It will be digested into the small pieces. Okay, separation of DNA fragments by electrophorese. Third steps, what you will do, through the electrophorese you have to separate the DNA molecules. Separate the DNA molecules like that. Separate the DNA molecules through the electrophoresis. This is the well where we load the DNA molecules. Okay. Just a minute. Now, separation by the electrophoresis. Next is the transferring blotting the separated DNA fragments to nylon sheet. Means from the gel DNA fragment transfer to the nylon sheet. This is called the southern blotting. If the RNA northern, if the protein western, if the protein eastern, not western. Protein will be western, not eastern. Protein will be western. Western blot. WESTERN. Western blot. So protein generally used for the digestion using labeled VNTR probe. Okay. Hybridization using labeled VNTR probe. Means probe we have already, and this probe already labeled with the VNTR, variable number of the tandem repeats. And last one is the detection of hybridized DNA fragments by the autoradiography. So with the help of autoradiography, we can detect the DNA fragments. Okay. Application of DNA fingerprinting. This is the very important. I told culprit you can find, and means culprit and rapist criminal, they can easily identified with the help of DNA fingerprinting. DNA fingerprinting comes in a branch of science which is known as the forensic science. This is known as the forensic science. This is called the forensic science. DNA fingerprinting belongs to the category of forensic science. Now, to solve the parental dispute, means parental dispute, I told one example, that is buffalo, means his mother and the child, we can identify easily. we can identify easily. It means this dispute easily identified. Okay, to solve immigrate problems. Okay, so peoples cannot migrate means to immigrate problems. If the some peoples they came from the different place. Okay, so there is the one is the immigration, and another is the immigration. Okay, immigration means the person which came from the any population and dissolve in our population. Are you getting or not? Immigration means some persons which came from the different population and enters in our population. That is called the immigrant. So we can identify this person belonging from the different country or different population. To identify dead bodies, soldiers died in wars. With the help of DNA fingerprinting, we can identify the dead bodies. Because DNA, we need only the DNA molecules. With the help of DNA, we can identify very easy. Now, to identify the dead bodies of person died as accident and bomb blast. If the person died into the accident and bomb blast, we can easily identify. Now, to identify the racial groups, means different person of the is different human race, we can identify with the help of DNA fingerprinting technology. To detect the inheritable disorder, I told already, metabolic inheritable disorder, SLR-S6 linked recessive disorder, autosomal dominant recessive disorder, autosomal recessive disorder. So we can find with the help of this DNA fingerprinting different types of the inheritable disorder. To detect the donor cell in case of the transplantation. If suppose that some person they have the given organ, so we can identify the person who have provided the organ, means donor cell we can identify when they have, with the help of DNA fingerprinting we can identify the donor cell in case of transplantation. Okay, and the replication, here they again, the single stranded DNA transfer to the nylon sheath using southern blotting technology, I told already. The single stranded DNA hybridized with the radioactive probes of the VNTR. The excess of probes are washed off. Southern blotting means when the DNA transfer from agar gel to nylon sheath, that is the southern blotting. And then next steps we have used to the probe hybridized with them, and some probe which is not clearly hybridized, it will be washed off. Okay, it will be the washed off. Nylon sheath is the X-ray photograph to get the bands of the VNTRs. We will see with the help of X-ray photographic plates, and we will get the bands of VNTR, variable number of tandem repeats. The bands of X-ray sheet is the DNA fingerprint. The bands which are generally observed on the DNA along with the VNTRs, this is called the DNA fingerprint. Comparing the DNA fingerprint with the sample and collected at the crime spot with the suspected identifies the individuality. Okay, I told comparing the DNA fingerprinting of the sample collected from the crime spot, that is the culprit. Okay, or where the accident or anything crime happened, with suspect identifies the individuality. So those person who did or who involves in this crime spots, we can identify with the help of DNA fingerprinting. Okay, southern blotting, I told very simple concept. The techniques transferring DNA from agar gel to the nylon sheet is called southern blotting. And probe single stranded polynucleotide fragment complementary to specific sequence nucleotide DNA, it is called the probe. And it is mainly used in the identify the VNTR, variable number of tandem repeats and desired scene. So probe is the very important thing. It is used for to identify the location of VNTR, variable number of tandem repeats and desired scenes. Okay, so with the help of this DNA fingerprinting, it breaks the hydrogen bonds. Okay, what it will do, DNA helicase enzyme breaks the hydrogen bonds, and in this way it helps in the unwinding of DNA molecules. Okay, so this is the very simple question, and now I'm going to finish. And tomorrow I will start new chapter, that is principle of inheritance and values. So thank you.